Hello everyone, my name is Malak Ibrahim Muhammad. I am a second year medical student, and today, inshallah, I will be giving you guys a past session on the role of B12 and folate in hemoglobin synthesis. This is a very simple lecture, so inshallah, I'm just going to try and make it as quick and simple as possible, and hopefully you guys can benefit from this, inshallah. This is my number. If you guys want to text me if you have any concerns after the lecture, you can always text me. And yeah, I also have my contact information, my email in the last slides. Okay. Bismillah. So these are the objectives that we're going to be covering today. We're going to go over an introduction to folate and B12, basically what they are, how they get absorbed, and how we can have a deficiency in these vitamins. We are also going to go over the role of B12 and folate in DNA replication and how this could result in megaloblastic anemia. We are also going to go over the clinical and laboratory findings of megaloblastic anemia, and we are going to have some practice questions in the end so that you guys can make sure that, solidify your information, make sure that you've gotten everything. The first thing that we're going to go over is the introduction to folate and B12. So what are B12 and folate? B12 and folate are essential water-soluble vitamins that are in our body, and because they're essential, we need to take them from outside the body. They are not produced, they're not synthesized inside of our body, so we need to consume them from multiple sources, which we're going to go over right now. We're going to start with B12. Another name for it is cobalamin. It is basically an essential water-soluble vitamin. As we said, it plays a critical role in a lot of functions in our body, like nerve function, DNA synthesis, and red blood cell production. It is mainly found in animal products such as meat, poultry, like fish, dairy and eggs, and fish. So we also have B9. Another name for it is folate or folic acid. Folate is the natural form, B12, and folic acid is the name for the synthetic form. You guys need to know these. They're very important because in questions they could ask you using the different names of these vitamins. They're not just going to say B9 and B12. They could use folate or folic acid. So you need to know the other names. So again, it is an essential water-soluble vitamin, and it is crucial for DNA synthesis and cell division. It is found in the liver, so in beef liver. It is very, very important from there. You also have leafy green vegetables, fruits, and fortified foods. Fortified foods are basically food products with nutrients added to them, like milk, which is fortified with vitamin D, bread, cereals, etc. So although both of these vitamins are water soluble, vitamin B12 can be stored in significant amounts for a long period of time. This is very important to know because if a person, for example, stops eating meat or fish and is a vegetarian, and they stop eating it for a year, they would still have stores of B12 because it's accumulated in the body. Basically, most of the time, it is primarily stored in the liver, while folate is stored in smaller amounts compared to B12. And this is why it is only sufficient for 4 months and requires more consistent dietary intake to prevent its deficiency. Okay, so this is a table that Dr. Abdul Jabbar put in your guys' slides. Honestly, you don't really need to worry about this at all. He even said it in the recording. You guys don't need to worry about the numbers. He just put this so you guys can have an idea of the sources of vitamin B12 and folate, which I've already mentioned in the previous slides. And honestly, that is all you need to know. So now we're going to go over the absorption of B12 and folate. So where do they get absorbed and how do they get absorbed? We're going to start with B12 or cobalamin. So absorption of B12 primarily happens in the ileum. What is the ileum? Basically, your small intestine, it has 3 parts. You have the C-shaped first part, which is the duodenum. You also have the jejunum, which is over here, and then you also have your ileum. Your ileum is the last part, the jejunum is the middle part, and the duodenum is the first part. So your B12, absorption of it primarily happens in the ileum. How does that happen? Your B12 is getting absorbed, it goes into your stomach, and this is where it is going to get carried by something called the intrinsic factor. Intrinsic factor carries this B12, binds to it, and it takes it to the ileum where it gets absorbed there. Now, what is intrinsic factor? It is basically a glycoprotein, which is produced by the parietal cells in your stomach. These parietal cells are located in the lining of the stomach, and they produce the intrinsic factor. This intrinsic factor, it binds to B12, and it carries it to the ileum, where it has receptors for this intrinsic factor. So the intrinsic factor is going to bind to this B12, and it's going to take it all the way to the ileum. And over there, you have receptors that would recognize the intrinsic factor, and this intrinsic factor would therefore facilitate the absorption of B12 at the ileum, because it has receptors for the intrinsic factor. So basically, if this intrinsic factor is not there, B12 will not get absorbed. This is so important. You guys need to know this. 100% you will get asked about it. Intrinsic factor is so important for the absorption of B12, okay? Now you have your B9 or your folic acid. The absorption of it happens in the jejunum. As we mentioned, this is the middle part of your intestine. The last part is called the ileum, middle part is called the jejunum. It is the middle section of the small intestine. And folate gets absorbed through active transport and passive diffusion. It doesn't need anything, to be honest. It doesn't need a glycoprotein like B12 does. And after this absorption, folate is converted to 5-methyl-tetrahydrofolate. And therefore, and over there, it is transported in the bloodstream. We're going to go a little bit into more detail onto that later, okay? Now, how does a deficiency in folate happen? A deficiency in folate and B12. A deficiency in these vitamins could result from a variety of causes, but mainly it is usually because of three main reasons. Number one is inadequate intake. So basically, since we mentioned that folate is found in leafy greens and fortified grains, a poor diet where you don't eat these leafy grains, like for example, you would see in vegetarians, not in vegetarians, people who don't eat meat. So a poor diet in these foods would lead to deficiency, especially in populations with limited access to fresh vegetables, right? So people, a lot of the people that would struggle with this would usually be older people. They will usually have this problem. This is inadequate intake. And then you have increased need. So people are pregnant, like in pregnancy and infancy, because they are not only supplying themselves, but they're also supplying another human being. So they will need, obviously, an increased amount of nutrition. Also, during periods of rapid growth, increase the body's demand for folate, and with convalescent patients. What are convalescent patients? Convalescence is basically the period during which a patient is recovering from a very serious injury or a surgery. So the body needs increased nutrients and resources in order to recover from the state, right? So that's increased need. And then a third reason for deficiency would be impaired absorption. Basically, you would see this in GI disorders. How would you see how would an impaired absorption result in deficiency? As we said, folate is absorbed in the jejunum, which is the upper part of the small intestine. After it is absorbed, it is converted and travels around the body as the form of 5-methyl-tetrahydrofolate, as we mentioned before. And impaired absorption of folate would result from gastrointestinal disorders. So if the jejunum, let's say it is under constant inflammation, like we would see in IBS, irritable bowel syndrome, there would not be able to absorb folate, and therefore it would result in a deficiency. So because the GI tract, the epithelium of it is inflamed, you would not be able to absorb it, and this is why you would have a deficiency in folate. This would also be seen in celiac disease or any conditions that would affect the jejunum, because this is where folate is absorbed. Now, anti-cancer drugs like methotrexate would also inhibit folate synthesis, leading to lower folate levels. Why does it inhibit folate synthesis? Because folate is crucial for synthesizing nucleotides, the building blocks of DNA, and we will see how that could result in megaloblastic anemia later on. But because folate is essential for synthesizing these nucleotides, we use that to our advantage. So cancer cells, because they rapidly divide and they require large amounts of nucleotides for their growth, if we inhibit this folate synthesis, the drugs would slow down the rapid growth and the division of these cancer cells. And therefore, we would be able to limit the growth of these cells. And this is why we use methotrexate. So obviously, that would result in a deficiency of folate. Okay. Now, deficiency in B12, it's mainly the same three things: inadequate intake. So since vitamin B12 is found in animal products, vegetarians and vegans are the ones who are at higher risk of the deficiency of this enzyme, unless they take fortified foods or supplements. The second thing is increased need. So again, pregnancy, infancy, all of the same things. And impaired absorption. But one more thing, we will add one more thing to impaired absorption over here, which is the deficiency in intrinsic factor. Now, B12, as we said, is carried by intrinsic factor in the stomach to the ileum, which is the last part of the small intestine, where it gets absorbed into the bloodstream. Now, if there is a deficiency in this intrinsic factor, like we would see pernicious anemia. Pernicious anemia is basically an autoimmune disorder in which the body attacks the stomach's parietal cells. And as we said, these parietal cells are the ones that make the intrinsic factor. So if your body is attacking these parietal cells, they would not be able to produce intrinsic factor, and therefore we would have a deficiency in them. And if we have a deficiency in intrinsic factor, we said that it cannot be carried all the way to the intestines where it will get absorbed. So this is why if we have a deficiency in intrinsic factor, vitamin B12 would not be able to get absorbed, which would lead to a deficiency. Okay, so this table, Dr. Al-Jabbar also put that for you. Honestly, we have mentioned everything that you need to know in the previous slides. So if you guys want to go over it, you will see pernicious anemia over here, which results in the abnormality of intrinsic factor or the deficiency. And honestly, if you want to go over it, you can, but you don't really need to. So checkpoint, does anyone have any questions for me? Is everything clear? Impaired absorption in folate. Yes, of course. So 1 second. Okay, so as we said, impaired absorption. Where, can anyone tell me where, as a recap, where does your folate get absorbed? Yes, in the jejunum, exactly. So your folate gets absorbed in the jejunum, right? So if you have some kind of syndrome in which the jejunum would be inflamed, it would not be able to absorb this folate. So GI disorders, in which the epithelium of the GI tract is inflamed, you would not be able to absorb this folate. So you would see this in irritable bowel disease or irritable bowel syndrome, IBS, or celiac disease. So anything that affects the jejunum itself or its absorption properties, it would result in an impaired absorption. That is number one. Number two, you would have anti-cancer drugs. How do anti-cancer drugs result in folate deficiency? It's because they inhibit folate synthesis. And they do that because we want to stop the division of these cells because they're proliferating quickly, right? And we want to be able to control that. We want to be able to stop that. And that is why we would give methotrexate, which would inhibit this folate synthesis, and therefore it would stop DNA replication. And this can obviously also lead to a folate insufficiency because you are stopping the synthesis of folate. Does this make sense? Is everything clear? Okay, perfect. Impaired absorption in B12, yeah, of course. So impaired absorption in B12, we said it's basically kind of the same thing, but as folate. So you would have a problem in GI disorders. So if you have GI disorders, anything that would affect the ileum, not the jejunum, because as we said, the jejunum is for the folate and B12 is for the ileum. A way that you guys can remember this is that 9 comes before 12. So you know how the jejunum is before the ileum. So 9 jejunum gets absorbed first before B12. Okay. So if there's any problem in the ileum, like if you have, let's say, ileal resection, right? You remove a part of the ileum, you would not be able to absorb B12 because this is where it gets absorbed. If you have Crohn's disease, anything that directly affects the ileum would result in a deficiency in B12. But another thing that differs from B12 and B9 is intrinsic factor. So if you have a deficiency in intrinsic factor, which is the glycoprotein that attaches it to B12 and carries it all the way from the stomach all the way to the ileum, if there's a problem in intrinsic factor or if you have a deficiency in it, without it, vitamin B12 would not get absorbed. Like there's no way for it to go through the bloodstream because intrinsic factor is the one that allows it to move there. Does this make sense? Is everything clear? Can I move on? Okay, perfect. So now we are going to be talking about the role of B12 and folate in DNA replication. This is the bulk of the lecture. This is the most important part. So please try and focus with me. Now, this diagram, I got it from Dr. Abdul-Jabbar's lecture. It's honestly really, really good. So I don't see a reason for me to use another one. So we're going to go over this. So the whole reason the B12 and folate are important in DNA replication is because they play a very important role in the formation of thymine. So thymine, can anyone tell me what thymine is? Of course, you guys know what thymine is. Yeah, it's a nucleotide in the DNA. So nitrogen is based in DNA, exactly. So thymine is important because it is the complementary nucleotide of adenine when we're making a DNA strand. So before we start, one thing that you guys need to know is that folate circulates around in our body. It circulates around in our body in the form of 5-methyl tetrahydrofolate. So folate does not circulate around the body in tetrahydrofolate or folate form. It circulates around the body in 5-methyl tetrahydrofolate. And as long as it's 5-methyl tetrahydrofolate, it is inactive. So it is just circulating around the body in the plasma, and whenever it is needed, it crosses the membrane and goes into the cytoplasm of the cell and gets converted into its active form, which is tetrahydrofolate. So tetrahydrofolate is the active form of folic acid, of folate. So 5-methyl tetrahydrofolate, it is not the it is the inactive form, and whenever you need it to be active, it gets converted to tetrahydrofolate. So in order to do this conversion from 5-methyl tetrahydrofolate to tetrahydrofolate, you will need to be able to give this methyl group over to something. You will need to transfer it somewhere. And who is responsible for accepting this methyl group? It is homocysteine. So homocysteine will take this methyl group for 5-methyl tetrahydrofolate, and in this process, it will become methionine. So 5-methyl tetrahydrofolate, it will give its methyl group, it will get transferred from here to homocysteine, giving us methionine. Who does this whole process? It is catalyzed by methionine, the enzyme that is responsible for it is methionine synthase. Methionine synthase is the one that is responsible for taking the methyl group from 5-methyl tetrahydrofolate to homocysteine, giving us methionine. But it cannot do this on its own. It needs a vitamin with it. It needs a cofactor. And this cofactor is vitamin B12. And this is why without vitamin B12, we would not be able to convert 5-methyl tetrahydrofolate to tetrahydrofolate. Is this part clear? Okay, I'll repeat it again. I'll repeat that again. Okay. So you have folate, and this folate is circulating around your body in the form of 5-methyl tetrahydrofolate. 5-methyl tetrahydrofolate is the inactive form. 5-methyl tetrahydrofolate is the inactive form. You cannot be able to use this as an active form if it's 5-methyl tetrahydrofolate. Whenever you need it, this 5-methyl tetrahydrofolate will get it will move into the cytoplasm, and this is where it will get converted to tetrahydrofolate, which is its active form. Okay. Now, in order to convert 5-methyl tetrahydrofolate to tetrahydrofolate, you will need to be able to give this methyl group over here to something. You will need to transfer it somewhere, and this transfer happens it is given to the homocysteine. So homocysteine, it takes or accepts this methyl group. It is transferred to homocysteine, and this transfer to homocysteine converts homocysteine to methionine. Who is responsible? What is the enzyme that does all of this, that takes the methyl group and gives this to homocysteine? It is methionine synthase. Methionine synthase needs help from something else. It cannot work on its own. It needs vitamin B12. So methionine synthase on its own cannot convert 5-methyl tetrahydrofolate to tetrahydrofolate. It needs a cofactor, which is vitamin B12. All right? So your tetrahydrofolate, now we have the tetrahydrofolate, and what are we going to do with this tetrahydrofolate? It is going to get converted into 5,10-methylene tetrahydrofolate. 5,10-methylene tetrahydrofolate, it does that using the amino acid serine. I need you guys to focus with me on this part. This part is important. So tetrahydrofolate gets converted to 5,10-methylene tetrahydrofolate because serine gets converted into glycine. It does that by giving it its methyl group, okay? So tetrahydrofolate requires serine, and the conversion of giving tetrahydrofolate to 5,10-methylene tetrahydrofolate requires serine. And the enzyme that does all of this is serine hydroxyl methyltransferase. Serine hydroxyl methyltransferase requires a cofactor, and this cofactor is your B6. B6 is the cofactor that you need in order for this enzyme to work. Without it, this whole process will not happen. Is that clear? Yeah, sure, I can repeat this part. So tetrahydrofolate, we convert it to 5,10-methylene tetrahydrofolate, and in order to do that, we need serine. Serine gives its methyl group to tetrahydrofolate, giving us 5,10-methylene tetrahydrofolate. Serine, after it does that, it becomes glycine. Okay, the enzyme that does this process, converting tetrahydrofolate to 5,10-methylene tetrahydrofolate, is serine hydroxymethyltransferase. Serine hydroxymethyltransferase requires a cofactor. What is this cofactor? B6. What is the cofactor that is needed for the conversion of 5-methyl tetrahydrofolate to tetrahydrofolate is B12. All right, so is that clear? Okay, perfect. So your 5,10-methylene tetrahydrofolate, what are we going to do with it now? Why do we need this 5,10-methylene tetrahydrofolate? This is what is going to be used to convert deoxyuridine monophosphate to deoxythymidine monophosphate. It does that using the enzyme thymidylate synthase. You can see number 3, thymidylate synthase. This enzyme is the one that converts DUMP, which is the short form of deoxyuridine monophosphate, into deoxythymidine monophosphate. Now, what is DUMP? This is basically the precursor of uracil. Can anyone tell me what uracil is? It's an RNA, basically. So it is the nitrogenous base that is instead of thymine that we would find in RNA. Now, DTMP, this is the precursor for thymine. Now, we cannot have thymine if this whole process doesn't happen. Basically, like we do all of this whole cycle in order to be able to convert folates or 5-methyl tetrahydrofolate into thymine, okay? So this 5,10-methylene tetrahydrofolate, it is going to allow thymidylate synthase to convert DUMP to DTMP, okay? So this forms deoxythymidine monophosphate, which is DTMP, and this DTMP gets converted into deoxythymidine triphosphate or DTTP. This then is used in RNA and DNA synthesis, okay? So this is the process in which we can convert folate into thymine. Can anyone recap this? Can anyone open their mic and recap this process or in text? Okay. Lean, can you open your mic?
Okay, sure. So first we have 5-methyl tetrahydrofolate. It's like circulating, and when we need it, it passes through the membrane, and then it converts to tetrahydrofolate. And how does this happen? Homocysteine takes the methyl group, I think, and it converts into methionine by the help of methionine synthase and vitamin B12. And then tetrahydrofolate converts into 5,10-methylene tetrahydrofolate. How does it do this? Serine, I think, adds a methyl group by turning it to glycine. And this happens through the enzyme serine hydroxymethyltransferase and B6. And then 5,10-methylene tetrahydrofolate converts the DUMP, which is a precursor of uracil, to DTMP, and then DTMP turns into thymidine, and then thymidine is used in DNA.
Yes, exactly. Good job. That is the perfect review, except for one thing that I want to make clear for you guys. Homocysteine isn't the one that takes the methyl group itself. Methionine synthase is the enzyme that takes this methyl group from 5-methyl tetrahydrofolate and gives it to homocysteine. Homocysteine, after it takes this methyl group, it becomes methionine. It doesn't take it itself. So the enzyme takes this methyl group and gives it to homocysteine, and this enzyme cannot do it on its own. It needs a cofactor, or it needs help, which is from the vitamin B12. Okay? All right. Any questions? We're going to recap this. One thing that I want to go over as well is that this 5,10-methylene tetrahydrofolate gets converted into dihydrofolate. This dihydrofolate, we have a very limited supply of tetrahydrofolate, and in order to replenish the supply, we can get it from two ways, basically. So the first way is we can convert this dihydrofolate into tetrahydrofolate, and this will replenish the tetrahydrofolate storage that we have inside the cell. And this is done by dihydrofolate reductase. Okay, these enzymes are important. Please remember them. But the most important thing that you need to focus on is at which part does which part is vitamin B12 is important, and at which part is vitamin B6 important, or folate. Okay, so the enzyme is dihydrofolate reductase. The second way that we can replenish this tetrahydrofolate storage inside the cell is by converting 5-methyl tetrahydrofolate back into tetrahydrofolate from the plasma, so taking it back in. But in order to do that, tetrahydrofolate on its own inside the cell, it needs to be bound to glutamate. So basically, this process is called polyglutamation. So THF inside the cytoplasm attaches to glutamate, and this process is important because it protects the tetrahydrofolate from hydrolysis or degradation in the body. And this prevents the efflux of tetrahydrofolate, it prevents it from getting outside the cell. It stays inside the cytoplasm and it doesn't get out by polyglutamation. This is important. Please do remember this. Okay. So as a recap, 5-methyl tetrahydrofolate to tetrahydrofolate and methionine. And this process requires the amino acid homocysteine because it is the one that takes the methyl group from 5-methyl tetrahydrofolate. And B12 is required as a cofactor. So 5,10-methyl tetrahydrofolate gets converted to DNA and rest get. No, it's not 5-methylene tetrahydrofolate that gets converted into thymidine. 5-methyl tetrahydrofolate gives its methyl group to DUMP, and this converts DUMP into DTMP. But 5-methylene tetrahydrofolate isn't the one that gets converted into thymidine. Okay, so 5-methylene tetrahydrofolate, after it gives its methyl group to DUMP, it becomes dihydrofolate. Is that clear? Okay, perfect. Now, tetrahydrofolate, it becomes 5,10-methylene tetrahydrofolate with the help of serine, and this serine becomes glycine. Now, this process requires the methyl that we said from serine and B6 as a cofactor. Now, DUMP, it gets converted into DTMP and DHF, or dihydrofolate, and this requires the methyl group from 5,10-methylene tetrahydrofolate, as we said, regenerating dihydrofolate. And dihydrofolate can go back into tetrahydrofolate by dihydrofolate reductase. Please tell me if this part is not clear. I can repeat it again. This is the most important part of the lecture, so I need to make sure you guys are clear on this. Can I move on? Okay. So how does this whole process relate to megaloblastic anemia? How is this relevant to us, and why do we need to know all of this? So one thing that I need you guys to know is that the first type of tissue that gets affected by any impairment at all in DNA synthesis are always the ones that are, do we need to learn the enzyme names? Yes, I believe they are important, so please make sure to know them. Can you please repeat the polyglutamation part? Yes, of course. So tetrahydrofolate, it needs to be bound to glutamate inside the cytoplasm. If it does not get bound to it, this tetrahydrofolate is susceptible to hydrolysis or getting damaged inside the cell, or it would leave the cell. So something needs to keep it inside the cytoplasm so it doesn't leave. And what is that? That is glutamate. So glutamate, it binds tetrahydrofolate, and this process is called polyglutamation. Polyglutamation basically just keeps THF storage inside the cell. It prevents it from going outside. Okay, is that clear? If it's confusing for you guys, after we're done with the session, I will go back to this to make sure that you guys got everything. Okay. So I'll repeat this again. So why do you guys think that this relates to megaloblastic anemia? It is because, as we said, the first type of tissue that gets affected in DNA synthesis are always the ones that are rapidly dividing, the tissue with the rapidly dividing cells, because these cells need to constantly replicate, right? So you need constant replication, and you need the DNA replication process to be, just working fine, no problems with it. And if any problems happen, you would see this with the tissue with the rapidly dividing cells. Where would you find this? You would find this in enterocytes, in skin, and hematopoietic tissue, which is what results in megaloblastic anemia. So I want you to imagine this with me. You have DNA polymerase, and it is replicating the DNA because the cell needs to divide, okay? It faces an adenine nucleotide, so it needs to complement it with a thymidine nucleotide. but it cannot find any because there's a problem with thymidine synthesis. So because of this problem in thymidine synthesis, it will not be able to complement adenine with a thymine because there are no thymine available. Now, instead of taking thymine, it takes uracil instead. By taking uracil, we have complemented adenine with uracil instead of thymine. And this is obviously not correct. And you have the DNA proofreading enzyme, right? So the DNA proofreading enzyme, it checks to make sure that everything is clear. And it notices that there is a uracil there, which is not supposed to be there. It's supposed to be a thymine. So what is it going to do? It will excise it out of the DNA strand. It will take it out and just throw it out, okay? What does this process do? It is supposed to remove it and replace it with a thymine, but it can't find any. So it just leaves a gap there in the DNA strand. And this strand results in a break in the DNA strand. This gap that is in the strand results in a break in the DNA strand. And when two breaks on opposite sides of the DNA, as you can see here, if you have two breaks on opposite sides of the strand, it will result to a fragmentation of the double-stranded DNA. I will tell you why this fragmentation is important to know and what is its significance. So when either folate or vitamin B12 is deficient, you will not be able to produce thymine. So the thymidine nucleotide synthesis is impaired. In diminished thymidine availability, uridine is incorporated into the DNA instead of thymine. And the repair process removes this uridine, leaving empty spaces in the replicated DNA, resulting in single-strand breaks. When excisions at opposing DNA strand sites coincide, the double-stranded DNA breaks occur. And repeated DNA strand breaks lead to fragmentation of the DNA strand. Now, this is a fragmented DNA, and this is a normal DNA strand, okay? So why is this significant to us? Why is DNA fragmentation a problem? It's because, so let's imagine this. We have an RBC, and the RBC, once it divides, it gets smaller and smaller with each division, right? So if there's a problem with this division, and every time the stem cell tries to divide, it needs to go through DNA replication. And if this DNA replication process is not there, and there's a problem with it, it would not be able to go through. It would just stay over there, unable to replicate. This results in the accumulation of these big blast cells that cannot divide. So the fewer the divisions, the bigger the cells. So if the less the cell divides, the bigger it is. And this is what results in megaloblastic anemia. How does fragmentation relate to this? It's because fragmentation disrupts the normal cell cycle by causing the erythrocyte progenitor cells to stay or stall in the S phase. They cannot go to the M phase, to the mitosis phase, right? They're just stuck there. They cannot divide. And because they cannot divide, the stalled replication process in erythrocyte progenitors leads to the formation of large immature red blood cells, which are known as megaloblasts. And these megaloblasts, it will accumulate inside the body, and it will not be able to divide. So you will have an accumulation of these megaloblastic cells that cannot divide, and this is what results into to megaloblastic anemia. So this fragmentation of DNA is non-functional. This fragmented DNA doesn't work, and it makes the DNA replication incomplete. And because the cell division stops, this results in apoptosis of these blast cells within the bone marrow. Now, because these blast cells are just there and they keep on accumulating, they result in megaloblastic anemia. Is that clear? Do you guys want me to repeat that? Okay, I'll repeat it. So, again, imagine this. Else, you have DNA polymerase, and it is replicating the DNA because the cell needs to divide. Every time you have a cell that needs to divide, it will need to go through the replication process. So the cell wants to divide, so it will start to replicate its DNA. It cannot replicate its DNA. Why? Because while the DNA polymerase is replicating the DNA, it faces an adenine. And the adenine, yes, there will be questions at the end of this lecture. Adenine, the complementary nucleotide for it is thymine. Now, because there is no thymine, this DNA polymerase will replace it with a uracil instead. This uracil is not supposed to be there, and adenine is supposed to get complemented with a thymine, not with a uracil. And who is going to notice that this is a problem? Your proofreading enzyme. The proofreading enzyme will go through the DNA strand. It will notice that there's a uracil that does not belong there. So it is going to excise it out of the DNA strand. It is going to remove it, and therefore, removing it, it's supposed to replace a thymine with it, the proper one that's supposed to be there. And because there's no thymine to replace it, it will just leave a gap in the strand. There will be nothing to replace it over there. And this gap, if we have another one on the other strand, you will have a fragmentation. If it coincides, if there are two strands with the same gap, you will have DNA fragmentation. So it is basically a break in the DNA strand because there is a problem, because there is insufficient amounts of a specific nucleotide, which results in a break in the DNA strand. Okay, now this break, it is important. Why? Because this break doesn't allow the cell to replicate. Now the cell will try to replicate, it can't, because it doesn't have enough nucleotides. And because it doesn't have enough nucleotides, it cannot divide and make a new cell. And so it just stays in this phase. It stays in this phase of the S phase, and it doesn't go into the M phase because it just can't. So the cell division stops, resulting in the cell lysis of this erythrocyte. And because it cannot divide, some other cells manage to go through this process and not go through apoptosis. So if they don't divide, they just stay big, because the more they divide, the smaller they get. And when they don't divide, they just stay as these megaloblastic cells. Blasts are basically progenitor erythrocytes, okay? So megablasts that cannot divide, they just accumulate inside the body, and they become megaloblastic, and they cause megaloblastic anemia. Is that clear? Yes, this happens in the bone marrow. Okay, can I move on? Okay, perfect. So we are going to go over the last thing in this lecture, which are the clinical symptoms and the laboratory findings. So the clinical symptoms of B12 and folate deficiency would be general symptoms of anemia, like you have weakness, shortness of breath, and pallor. You would also see patients who show mild jaundice. Can anyone tell me why you would see mild jaundice in these patients with B12 and folate deficiency, or patients with megaloblastic anemia? Just a guess. Not enough iron? No. So remember how we said bilirubin? Yes, exactly. Perfect. Great job. Accumulation of bilirubin. Yes, exactly. Great job, guys. So basically, when a red blood cell, when a hemolysis happens, which is basically the breakdown or the rupture of this RBC, when it happens, the content inside of it will go out. And this content is hemoglobin. This hemoglobin becomes bilirubin. And usually, this bilirubin can be metabolized by the liver. So as we said, can be metabolized by the liver. But the liver, because there's too much bilirubin, it cannot metabolize all of this bilirubin, so it accumulates inside the cell tissue, okay? So that is why we would have mild jaundice in B12 and folate deficiency. You would also have epithelial damage that affects the protective and absorptive functions of the GI tract, causing gastritis, nausea, and constipation. Gastritis is basically just the inflammation of the lining of the stomach. So in megaloblastic anemia, I'm going to repeat the jaundice part. So in megaloblastic anemia, your RBCs, they are large and fragile, okay? They are abnormally large and fragile. And these cells, they break down prematurely in the bone marrow due to the ineffective erythropoiesis within the circulation, okay? So this leads to increased destruction of these RBCs. As the RBCs break down, hemoglobin is released and metabolized into bilirubin. Usually the liver processes this and clears out this bilirubin, but in this case, as we said, the amount of bilirubin produced exceeds the liver's capacity to clear it, which leads to the buildup of this bilirubin inside the tissues and causing this yellow hue in the skin and the eyes, right? You also have glossitis, which is one of the main symptoms that you would see with B12 and folate deficiency. Glossitis is basically caused by the atrophy of the epithelial cells on the tongue. And as we said, one of the main tissues that would be the first tissues that would be affected by any problems or any impairments in DNA synthesis or DNA replication would be the tissues that have cells that replicate quickly. And these include epithelial cells that are located on the tongue. And because they cannot keep up and keep on replicating, they can't replicate anymore, this results in the inflammation and the loss of the normal tongue structure. All right, is this clear? I'll move on. So what are the lab findings that you would see with B12 and folate deficiency? You have the complete blood cell count findings. You would see macrocytic anemia, and that would be noticed because of the high mean cell volume, which is normally more than 98 femtoliters, and it could get as high as 120 to 140. This reticulocyte count is low. You would also see a reticulocyte count that is low, and this reticulocyte count suggests that there's inadequate bone marrow response to the anemia. Okay, and then you would also see total white cell and platelet counts. They would be reduced, and you would see this especially in severe anemia. Okay. Blood smear and bone marrow findings, you would see hypersegmented neutrophils, which are basically neutrophils with more than 5 lobes, and this is a classic finding in megaloblastic anemia. You would also see that the bone marrow is hypercellular and the erythroblasts are large. You would also see in the vitamin and serum levels, you would see that serum unconjugated bilirubin is high, and we explained why that happens. And you would also see high levels of lactate dehydrogenase. Lactate dehydrogenase is basically as an enzyme that is stored in the cytoplasm of all cells, including RBCs. So when an RBC ruptures, this LDH will get released into the bloodstream, and therefore we use it as an indicator of cell damage or tissue damage and hemolysis. Okay, so that is it. Checkpoint. Any questions? We'll move on to the questions now. You want me to repeat the very last part? So vitamin and serum levels, you would see that bilirubin is increased because when the RBCs, they rupture, this bilirubin is going to get released into the bloodstream, okay? You would also have lactate dehydrogenase, which is basically an enzyme that is located in all cells. It is located in the cytoplasm of all cells, including red blood cells. When this red blood cell ruptures, these the LDH, this enzyme, would get released into the bloodstream, and therefore you would have an increased level of it in the blood. So this is why they use LDH, or lactate dehydrogenase enzyme. They use LDH levels to check whether there is, they use it as an indicator of tissue damage and hemolysis. So if there's increased hemolysis, you will see an increase in lactate dehydrogenase because the RBCs are constantly lysing. Is that all good? Perfect. So onto the practice questions. We'll start with the first question. So I'll give you guys a minute to answer this. Actually, I'll give you guys 35 minutes. 35 seconds. Please answer it in private so other people get a chance to answer. Okay, does anyone want to open their mic and answer this? I'm getting E and A in the chats. Okay. So does anyone want to read it and answer it? Okay, go ahead, Lean.
Okay, so a 34-year-old man has schizophrenia and has had chronic fatigue for 6 months. He has a good appetite, but has refused to eat vegetables for 1 year because he hears voices saying that vegetables are poisoned. His physical and neurological examinations are normal. His hemoglobin level is 9.1. Leukocyte count is 10,000 and MCV is 122, which of following is the most likely diagnosis. So MCV is macrocytic, so it's either B12 or folic.
Yes.
And also, because he refused to eat vegetables, I'm pretty sure that folate comes from vegetables.
Yes,
So, A.
yes, exactly. Good job. So if we go back, this is the answer. The answer is A. So as Lean said, because they are not consuming veggies, which is where we get our folate from. I'll go back to the slide so you guys can check. Here, you can see that you get folate from leafy green vegetables, fruits, and fortified foods. And this is why, because they were not consuming vegetables, this is why they had a deficiency in folate and not in B12. Although his lack of vegetable consumption could suggest either vitamin B12 or folic acid deficiency, another thing that I mentioned to you guys is that the body has sufficient stores of vitamin B12 that could last us from 4 to 5 years. So even though he hasn't eaten veggies in a year, he still has a storage that would last him up until 3 years. So he does not have a vitamin B12 deficiency because of that. The body only stores folic acid from 3 to 6 months, and this is why 1 year of not eating veggies would result in a complete folic acid deficiency. Okay, next question. Okay, so I'm getting a lot of different answers. I'm getting A's, B's, and E's. Okay, does anyone want to explain in chat why they chose their answer? Okay. I'm just going to explain this one. So the answer is actually B. So I will go back to the diagram to show you guys, but A is wrong because it's not the biosynthesis of pure nucleotides, but actually the biosynthesis of pyrimidine, because you are making thymine, right, which is a pyrimidine, and this is why A is wrong. Now B, it is involved in the conversion of 5-methyl tetrahydrofolate to tetrahydrofolate. If we go back, okay. So as you can see here, 5-methyl tetrahydrofolate gets converted to tetrahydrofolate using the enzyme methionine synthase. And as we said, methionine synthase requires B12 as a cofactor. And this is why vitamin B12 is responsible or important in the conversion of 5-methyl tetrahydrofolate to tetrahydrofolate. Please make sure you remember this diagram, guys. It's so important. Okay, let's go back to the question. Okay, hold on. Is this clear? Does anyone want to ask any questions about this? Okay, I'll move on. So this is the third question. I'll give you guys a few seconds to answer this. Okay, perfect. Everyone is answering it correctly. It is A. Polyglutamylation is basically the process of binding tetrahydrofolate to glutamate. And this process is what keeps the tetrahydrofolate from getting out of the cell. So it prevents its efflux out of the cell and it keeps it inside the cytoplasm and it protects it. So this process is very important to trap fully inside the cell. Last question. Yeah, sure. I'll show you the picture after. Yeah, there is. Hold on. Okay. So if we look here, you have tetrahydrofolate, and as we said, tetrahydrofolate, in order to keep it inside the cytoplasm, preventing it from going outside, you have a process called polyglutamation. Polyglutamation is important because it prevents tetrahydrofolate from going out of the cell. It stays inside the cell, and this is how we replenish tetrahydrofolate, because every time we would convert dihydrofolate into tetrahydrofolate, it could just like pick for the question. Oh, my bad. Hold on. Okay. Can you see it now? My bad. Sorry, guys. Okay, that's the answer. Okay, I'll just answer this. So, a 65-year-old woman presents with tingling sensation in her lower limbs for the last 2 months, and a complete blood count shows low hemoglobin, and the peripheral blood smear shows the attached picture. So, as you guys can see, we have the polynuclear neutrophil, and it has a lot of lobes. So polymorphonuclear neutrophil, which will result in vitamin B12, which is a sign of vitamin B12 deficiency in the lab findings. And that is it. Do you guys want me to go back and repeat any of the concepts I've explained? Yes, I can go back to the diagram and explain it again. Okay. So the first thing that you need to know is that 5-methyl tetrahydrofolate is the form of folate that travels around the plasma. Now, when we need this 5-methyl tetrahydrofolate, it is going to go inside the cell through the plasma membrane, and it is going to go inside the cytoplasm. This 5-methyl tetrahydrofolate, it will get converted into tetrahydrofolate. Now, this 5-methyl tetrahydrofolate to tetrahydrofolate conversion, how does it happen? By the enzyme methionine synthase. Methionine synthase will take the methyl group from 5-methyl tetrahydrofolate and will give it to homocysteine. Homocysteine, after it takes this methyl group, it will become methionine, okay? Now, methionine synthase, it requires a cofactor or a help. It needs help from another vitamin. Now, this vitamin is vitamin B12. So vitamin B12 is the cofactor for methionine synthase, which takes the methyl group from 5-methyl tetrahydrofolate and gives it to homocysteine, giving you tetrahydrofolate, okay? Now, this tetrahydrofolate, it will get converted into 5,10-methylene tetrahydrofolate. How is this going to happen? By the help of serine and the enzyme serine hydroxymethyltransferase. It will take the methyl group from serine and it will give it to tetrahydrofolate. This tetrahydrofolate, after it takes the methyl group from serine, it will become glycine, and tetrahydrofolate will become 5,10-methylene tetrahydrofolate, okay? The enzyme that is responsible for this process is called serine hydroxyl methyltransferase. Serine hydroxyl methyltransferase requires a cofactor, and this cofactor is B6, okay? Now, 5,10-methylene tetrahydrofolate, it will take this methyl group that it has. The enzyme that will take it is thymedylate synthase. Thymedylate synthase will take the methyl group from 5,10-methylene tetrahydrofolate and it will give it to DUMP. DUMP will get converted into DTMP after it takes this methyl group. DTMP then gets converted into DTP, which is then used to make thymine and is then used in DNA replication. Now, after 5,10-methylene tetrahydrofolate, it gives its methyl group, it becomes dihydrofolate. Dihydrofolate gets replenished. We need to replenish our tetrahydrofolate stores. And in order to do that, we need to convert the dihydrofolate that we have back into tetrahydrofolate using the enzyme dihydrofolate reductase. Dihydrofolate takes this dihydrofolate reductase takes this dihydrofolate and converts it into tetrahydrofolate. And this is how we replenish the tetrahydrofolate storage inside the cell, okay? This tetrahydrofolate, in order to keep it inside the cytoplasm and not to to prevent it from going out of the cell, an efflux of tetrahydrofolate, we need to bind it to glutamate, which keeps it inside the cytoplasm. And this process is called polyglutamation. Is that clear? Yes, when we say B12, it acts as a helper to the enzyme. And this enzyme is methionine synthase. Okay. Thank you so much for attending, guys. I hope this was beneficial for you. If you have any questions for me, please do text me. I have my number here, I have my email, and I wish you the best of luck. Thank you.